Hi guys, my name is Venkatnag Sai Raju Alluri, or Sai for short. I'll be doing a presentation on restriction enzymes. Before we get into this presentation, I'd like to give a shout out to my sponsor. Wait, I don't have a sponsor because I literally have one subscriber. So let's just get back to the presentation, shall we? So my contents are the following, as you can see. I'll be starting off with the history of restriction enzymes. So essentially, in the early 1950s, experiments for two teams of researchers, that is, Salvador Luria with Mary Human, Joe Bertani working with Jean Weigel, showed that some strains of bacteria were more resistant to viral infections than others. Next, in the 1960s, Werner Arber observed a dramatic change in the bacteriophage DNA after it invaded those resistant strains of bacteria. So essentially, it was degraded and cut into pieces where in an attempt to explain the resistance of certain bacterial strains to the bacteriophage infection. Arber then posited this idea that bacteriophage resistant bacterial cells might indeed express a specific enzyme that degrades only invading bacteriophage DNA, but not their own DNA. A few years later, in 1970, Hamilton Smith not only independently verified, Arber's hypothesis, but also elaborated on this initial discovery by Lin and Arber. So, based on this, he was able to successfully purify a restriction enzyme from another bacterium, which is Haemophilus influenzae, and definitely showed that this enzyme cut DNA in the center of a specific 6-base pair sequence. So it's very, it's quite lengthy, but it is very pertinent to get this, out of the way when we go into the introduction. So as the aforementioned developments gave a clear-cut definition about restriction enzymes, we can define this by stating that they're also called restriction endonucleases, and that they recognize a specific sequence of nucleotides in double-stranded DNA and cut the DNA at a specific location. So on the right, you can see an image of a very simplified version of the process of, the use and process of restriction enzymes. Here you can see the cut site along with the cut site mutated and how it, where the gels are run, giving us our final product for the required processes. Going into the RE nomenclature, we are given a few points here. For example, the enzyme HIND3 was isolated from Haemophilus influenzae strain RD. What we need to understand here is that the first three letters of the name are italicized because they abbreviate the genus and species names of the organism. The fourth letter typically comes from the bacterial strain designation. The Roman numerals are used to identify specific enzymes from the bacteria that contain multiple restriction enzymes, as well as the order of isolation. So the types of restriction enzymes and classification, the more narrow division is not something we do not know. We have the exonucleases and endonucleases, but what's more pertinent is the classification into three main categories, which involves type 1, type 2, and type 3. As for type 1, it recognizes specific DNA sequences, but makes sure that they're cut at seemingly random sites that can be as far as 1,000 base pairs away from the recognition site. So you have an example of ECO-B and ECO-K. Type 2, which recognizes and cuts directly within the recognition site. You have HIND2 and HIND3. As for the type 3, it recognizes specific sequences but makes their cut at a different specific location that is usually within 25 base pairs of the recognition site. Here's an obvious chart that we usually see around the web. It gives a list of various properties and parameters that we look into when discussing various types and categories of the restriction enzyme. So you can pause the video and look into this. So as for the applications, we have, though there are many, we have essentially three major important applications. Those involves genetic engineering, DNA mapping, and gene sequencing. And other applications which are somewhat miscellaneous compared to the above mentioned three are gene expression and mutation studies, which again through proper application bolsters the research and development and application obviously for the above three. But coming to something that's not so known, or rather being looked into, you have the future R&D for broad application. So in this regard, we have restriction enzymes as a target for DNA-based sensing and structural rearrangement. So we need to understand that it has broken the constraints of a traditional restriction enzyme technology. Okay, it is quite revolutionary, and we'll see why very briefly, of course. DNA nanostructures have been shown to be viable for the creation of a complex logic-enabled sense motives. To date, most of these types of devices have been limited to the interaction with strictly DNA type inputs. So we have this new hypothesis that aims to integrate with the DNA structure for use as inputs to trigger structural transformation and structural rearrangement. So this hypothesis is available in the given bibliography, so everyone can check out the current research that's going on, and perhaps if you're a student or a professor or whomever, you can contribute to that. So thank you all for listening. Thank you all for listening, and I hope you have a good day. But don't forget to like, share, and subscribe.